Hi class, welcome to the Module 13 and 14 of our Biochemistry for Medical Laboratory Science. And for today's lecture, we will be discussing the amino acid and as well as the urea cycle. So nandito na po tayo sa part ng biochemistry wherein pag-aaralan na natin kung saan napupunta yung mga waste products of our biomolecules. So for the past weeks, nade-discuss na natin yung mga pathways involved in the biomolecules. Pero dito naman po sa week na to, starting this week, puro yung mga waste products na nila and then kung ano na yung nangyayari after the pathway, okay, after the breakdown of such molecules. Hence, the module 13 and 14 will be about the amino acid and then the urea cycle. So to give you a brief introduction of our protein or your amino acids, it is said that the metabolism of your amino acid comprises a wide array of synthetic and degradative reactions which amino acids are assembled as precursors of your polypeptides or other compounds and broken down to recover metabolic energy. The chemical transformations of amino acid are distinct from those of your sugar and then your lipid because merong nitrogen na involved. Again, it is distinct and it is different from other biomolecules because it contains nitrogen. This is particularly talking about your proteins. What else? Your amino acids, these are monomers of your, these are monomers, and when you combine small monomers and then it will build up such what we call polymers, yun yung tinatawag natin na mga proteins natin. Okay? Again, balikan lang natin building blocks of your proteins, amino acids. What else? Bulk of your cells' amino acid are incorporated into the protein. Every day, we synthesize protein depending on the diet, and then yung katawan natin, meron din tayong ability to synthesize those proteins. And then, madalas din nagde-degrade din sila. What else? Your amino acids, wala pong storage form. No storage form. This is for your amino acid. Sorry for the handwriting. Excess dietary amino acid are not simply excreted, but are converted to common metabolite precursors. Yung mga ano natin yan, yung mga sugar, fatty acids, yung mga ketone bodies natin, and then they can be used as metabolic fuel. What else? Ano pa bang mga statements natin regarding proteins? There are no enzymes. Okay, pakitandaan, there are no enzymes that will act upon your proteins in the mouth. Yung mga enzymes natin na magsa-start ng catabolism, ng pagbe-breakdown ng ating mga tinatawag na proteins, it will start on your stomach. Again, ang ating bibig, ang ating mouth, may mga enzymes po dyan kung saan pwede na mag-start yung pag-breakdown ng ating mga intake na food. Sa mga sugar, applicable sila. But for your proteins, there are no enzymes that will act upon your proteins in the mouth. The catabolism or the breakdown of the swallowing food will begin on your stomach and will end sa small intestine. Natatandaan sa inyong mga intestine, sa ating mga intestinal epithelial cells, nakakapag-generate sila ng mga tinatawag nating enzymes, mga pepsin, trypsin, yan. Diyan po sila lahat na-generate. Also, saan ba natin ginagamit yung mga amino acids natin? Bakit ba sila pinag-aaralan at bakit sila importante? Most important functions of your amino acids is that they are used to synthesize the proteins that we need in the body. Without these amino acids, wala po tayong proteins. So, the deemed importance of your amino acid is it is used as for the synthesis of your proteins that we need in the body. So ano-ano ba yung mga proteins na pwede natin makita sa body? So almost lahat yan. Mga enzymes, hemoglobin, myoglobin, mga plasma proteins natin, mga collagen, keratin, at lalong-lalo na yung mga enzymes. Okay? After synthesizing the proteins that are needed in the body, amino acids are used to synthesize yung mga tinatawag nating essential non-protein nitrogenous substances, which will be discussed sa susunod na slide. But again, without these amino acids, wala tayong mga proteins. So let us talk about the protein turnover. It refers to the balance between the proteins undergoing breakdown or degradation and the proteins being synthesized. So hindi po pwede na patuloy lang tayo nagsisynthesize or gumagawa ng mga amino acids sa katawan na hindi without going these molecules into a breakdown. Okay, hindi lang tao yung nagbe-breakdown, also yung mga biomolecules natin. Kasi ang mangyayari, magiging madami yung supply nila sa katawan natin, which is not good na din naman kasi dapat everything is in balance. Every day, there are proteins in our body that undergo breakdown. Also, there are also proteins that are being synthesized every day. This totals to 1 to 2%, about 200 to 300 grams of the total body proteins. And then every day you change 200 to 300 grams of your proteins. When proteins undergo breakdown, hindi lang tao nagbe-breakdown, pati sila proteins, the products are your amino acids. Again, ang pinaka-product ng protein breakdown is what we call amino acid. Kasi nga naman, is polymer siya. Pag binbreak mo siya into pieces, magiging amino acid siya. And then the 75 to 80% will be recycled and used again for protein synthesis. And then the 20 to 25% will be broken down to urea. Again, ano po ang urea? Bakit kanina pa binabanggit ni ma'am? Urea represents the final end product of the protein metabolism. Again, it is the final end product of protein metabolism. Ma'am, bakit sabi mo product po ng protein breakdown si amino acid? Yes, it is a product, but urea, it represents the final end product of your protein metabolism. Then it is excreted in the urine. Okay? The more protein that you eat, the more will be your urea excretion. Then the less the protein you eat, the less will be your urea excretion. So para silang may direct relationship, proportional sila. Okay, baka mahilig ko na naman itanong sa exam, pakitandaan na po. So for the high protein diet, yung mga mahilig uminom ng whey protein dyan, yung mga nag-gym, the higher the urea. Okay, ibig sabihin nun, mas madami yung in-intake mo na protein, depending kung kinain mo siya or incorporated siya sa iyong drink or incorporated sa iyong meal, you have ate a high protein diet, the more will be your urea excretion. And then, the higher the volume of the urine, ibig sabihin nun kapag ka parang polyuric ka, madami yung volume ng urine mo, urea kasi diuretic yan, diuretic. Okay, so the volume of urine to be excreted will also go up. So mas madami yung intake mo ng protein, mas madami yung urea mo, mas madami yung iiihi mo. And then, lastly, kapag high protein diet ka, naka-high protein diet ka, mas mataas din yung urine specific gravity. Ma'am, bakit narinig ko na si urine specific gravity? Yes, isa po siya sa mga minimeasure doon sa ating urine strips, kasama siya. Okay, doon sa urine strips na minimeasure natin aside from the pH, ayan, kasama siya sa chemical testing of your urine. Kasama sa pH, specific gravity, sila albumin. Ayan, kasama po sila doon. Urea is solid. It will also increase the specific gravity of your urine. So, tandaan po yung mga yan. Okay? These are all incorporated with the high protein diet. The higher the protein, the higher the urea, the higher the volume output or the urine volume output. So next, these are the overall sources and utilization of your amino acids, particularly amino acids ang pinag-uusapan natin. So again, our body patuloy na nagsisynthesize yan ng mga proteins. Okay? Depende yung sa intake ng ating dietary protein, pwedeng galing sa food na kinakain natin or pwedeng galing talaga naturally from our body. So ito yun. Source, again, source. Okay? And then it will be broken down into simpler form or what we call amino acids. And then these amino acids will be broken down pa or meron na siya mga waste products. Pag nag-undergo siya ng isang pathway, pwedeng maging urea, pwedeng maging ammonia, it can be glucose, ketones, and carbon dioxide. What else? It will go transamination. Transamination, it is removing the nitrogen from your amino acid. Okay, involved din po siya sa glycolysis and Krebs cycle. And lastly, yung mga amino acids natin, pagka hindi sila nagamit, pwede silang, or ito yung mga derivatives nila, yung mga tinatawag na non-protein derivatives. Okay? Essential non-protein nitrogenous substances, these are non-proteins, but they require amino acids for their synthesis. Again, yung nandito sa right side, yung non-protein derivatives, itong mga nandito sa right side, these are essential non-protein nitrogenous substances. There are non-proteins, okay, these are non-proteins, all of them, but they require amino acids for their synthesis. Hindi kagaya ng mga ibang molecules or substances, they'll be needing amino acid or they'll be needing amino acid and they are protein. But however, itong mga nasa right side, hindi sila hindi sila protein pero kailangan nila ng amino acid for their synthesis. So itong mga non-protein derivatives lang to. Pero itong mga nasa right side, yung mga sinasabi nating non-protein nitrogenous substances. Isa-isahin natin sila, si N1. Okay, purines po yung mga yan. Si N1, it comes from aspartic acid. Sorry for the handwriting. Si C4, C5, and N7 comes from glycine. Yan, mga amino acids natin. Yung N3 and N9 they come from glutamine. Next, yung porphyrin, ma'am, narinig ko na yan they require glycine pa rin naman. Next, si folic acid, narinig nyo na rin to, they require glutamic acid. Ito yung mga requirement nilang amino acid, kagaya na sinabi natin dito. Next, si creatine, tripeptide, they'll be needing GAM, G-A-M. Ano yung GAM? Glycine, arginine, methionine. Again, GAM. Glycine, arginine, methionine. Next, si glutathione, tripeptide shot. Ibig sabihin, tatlo yung kailangan niyang peptides. Si GCG. GCG. It needs glycine, cysteine, and glutamic acid for its synthesis. Okay, again, ulitin lang natin yung overall sources of and the utilization of your amino acids. Our body every day nakakapag-produce tayo or nakakapag-synthesize tayo ng ating mga proteins depending or ng amino acids depending na lang sa ating food intake or doon sa naturally occurring amino acids natin. And then, it will be broken down to its weights, mga products. And then, it can be converted to non-protein derivatives. Okay? Sa right side naman natin, ito naman yung mga tinatawag nating essential non-protein nitrogenous substances. These are non-proteins but they require amino acids for their synthesis. Please do take note that if an amino acid is not used, it will undergo catabolism or breakdown. Pag sinabing catabolism, breaking down of a molecule or a substance. So these are the two major pathways wherein the degraded intracellular proteins happens in the eukaryotic cells. Hindi na natin siya i-elaborate, but we have to know that it has two major pathways, the lysosomal degradation as well as the ubiquitin. And how do you pronounce that one? Hindi ko sure. Again, two major pathways, the lysosomal degradation and the ubiquitin. So next, pag-aralan natin yung pinaka-chemical structure of your amino acid and how it goes doon sa kanyang pinaka-breaking down. Okay? Ipaparte-parte kasi siya dito sa ating discussion. So mahalaga na alam natin yung basic ng chemical structure bago siya i-break down or i-broken down into pieces. Okay, tandaan, itong nandito sa ating left side, si amino group, NH2. Okay, it can be converted to waste and then it can be reused again by the body. Next, si R group, di ba? Nandito yung kanilang main difference, yung mga 20 amino acids natin. They are all the same doon sa ibang chemical structure. Nagkaiba-iba lang sila sa R group. R side chain, parang tama ba? And then the carboxyl group, it has a carboxyl group. And then yung pinaka-carbon. Okay? An amino acid has three reactive parts. We have your amino group, the carboxyl group, and then the R group. Huwag na natin pansinin si alpha carbon dyan. Okay? Pansinin na lang si amino group, si carboxyl, and si R group. And then this part will go oxidation. Okay? Pag-uusapan na po natin, nandito na tayo sa parte ng discussion kung saan, ano nang mangyayari doon sa mga pinaka-simplest form. Ma'am, sabi niyo hindi siya nabe-breakdown kasi simplest form na siya. Nabe-breakdown pa din naman siya, hindi into parang chemically structure niya. Pero amino acid as monomer, hindi na. Wala na siyang mas monomer pa. Pero kung i-breakdown siya into chemical pieces na, yung chemical structure na niya, yung mga functional group na niya, yung hihiwa-hiwalayin natin. Okay? So this is the amino acid catabolism. amino acid catabolism, paki-focus. The removal of the alpha amino group, meron siyang tatlong example or 3 processes. Yung alpha amino yung pinag-uusapan dito ha. Ito yung i-breakdown natin. We have the oxidative deamination, the non-oxidative deamination, and transamination. Again, it is the removing or the removal of alpha amino group. We have 3 processes. It involves the oxidative deamination, the non-oxidative deamination, and transamination. Ang pinag-uusapan dito, removal of alpha amino group. Ano po yun? Ito. Okay, next. Meron naman tayong tinatawag na decarboxylation, oxygenation, and 1-carbon transfer. Hindi na po sila involved doon. Iba na sila, ibang processes na. Basta po sa alpha amino group, yung 3 lang na yun. Okay? So, discuss natin siya. Accordingly, unahin natin si letter A, removal of alpha amino group. The removal of alpha amino acid, oxidative deamination muna tayo. The overall reaction is your amino acid will be converted to alpha keto acid plus ammonia. And then what are the enzymes involved? Glutamate dehydrogenase reaction, number 1. Number 2, the L-amino acid oxidase. And then yung mga reaction involved nandoon sa ibaba per enzyme. So sabi nga po natin, very, very important si enzymes when it comes to the biological function of a eukaryotic cell, tayong mga humans. Okay, almost lahat ng mga biological functions happening inside our body, meron kaakibat na enzymes yan. They serve as catalyst. Next, the D-amino acid oxidase. Okay, so for the oxidative deamination that is under the removal of alpha amino acid, we have 3 enzymes involved, and naming your glutamate dehydrogenase reaction, the L-amino acid oxidase, and the D-amino acid oxidase. So, rundown natin sila isa-isa. Pero hanggang ano lang siya. Yung dalawa lang. So the glutamate dehydrogenase, it is the only known enzyme in at least some organisms. It can accept either NAD+ or NADP+ as its redox reaction coenzyme. It is regulated allosterically by prenucleotides. When there is needed for oxidation of amino acids for energy, the activity is increased in the direction of glutamate degradation by ADP and GTP, which are indicative of low cellular energy level. Your GTP and ATP, indicative of an ample energy level, are allosteric activators in the direction of glutamate synthesis. Next po, for the D-amino acid oxidase, it occurs mainly in the kidneys. Its function is an enigma since D-amino acids are associated mostly with bacterial cell walls. Very little of D-amino acid isomer is found in humans, and then the role of D-amino acid oxidase may be in degradation of D-amino acids derived from intestinal bacteria. So ito most likely nakikita to sa ating mga prokaryotic cells. Okay, but nabanggit lang siya for this slide. So next naman si non-oxidative deamination, and then these are the 2 enzymes that are involved for the non-oxidative. We have your amino acid dehydrase and then the amino acid desulfhydrase. Okay, so dalawa lang siya. For the oxidative, we have 3. So next, the transamination. Nasaan po si transamination? Nandito po. Okay, removal pa rin po tayo ng alpha amino group under the amino acid catabolism. Baka kasi nalilito. Okay, for the transamination, balikan ko lang. It is the major process of removing nitrogen from amino acids. Ma'am, ano ulit? It is the major process of removing nitrogen from amino acids. In most instances, the nitrogen transferred as an amino group from the original amino acid to alpha-ketoglutarate. It is considered as the most active amino group as acceptor. Again, yung alpha-ketoglutarate considered as the most active amino group acceptor. Then we'll convert it to glutamic acid, while the original amino acid is converted to its corresponding alpha-keto acid. All amino acids can undergo transamination reactions except these 3, si lysine, proline, and threonine. LPT, PTL. Okay, pag tinanong sa exam, does all of your amino acids undergo the transamination reaction? No. Bakit? Kasi we have these 3 amino acids na exception to the rule, that are exception to the rule. Okay, tatandaan po ha, yung tatlong pinapatandaan ko, si PTL, accepted sila for the transamination process. What is again the transamination process? The removal of the nitrogen in the amino acids. Okay, so let us proceed. So, meron pa rin naman pong mga enzymes involved, hindi lang doon sa mga first 2 processes na nabanggit natin. Again, kada lahat nga naman ng mga processes and biological functions na nangyayari sa katawan, parati namang may involved na enzyme. Mahabang pangalan ng enzyme. So, for the transamination, meron tayong tinatawag na transaminases or the aminotransferases. So to carry the amino group of your amino acid, aminotransferases require participation of aldehyde-containing coenzyme pyridoxal 5-phosphate or your PLP. Ang haba, ma'am. It is a derivative of your pyridoxine or your vitamin B6. Most aminotransferases accept only alpha-ketoglutarate to a lesser extent. Oxaloacetate as the alpha-keto acid substrate in the second stage of the reaction, thereby yielding glutamate or aspartate as their only amino acid products. Again, yielding only glutamate or aspartate as their only amino acid products. The amino groups of most amino acids are consequently funneled into the formation of glutamate or aspartate, which are themselves interconverted by glutamate aspartate aminotransferase. So ito siya basically yung pinaka-reaction niya. Okay, the glutamate plus oxaloacetate is converted to alpha-ketoglutarate and then the aspartate. So ano ba yung goal nung mga nabanggit nating processes to remove the alpha amino group? Ang goal lang naman po natin is to eliminate the ammonia. As we all know, ammonia is very toxic in the body. Okay? Ammonia, ano po ba yung chemical structure niyan? Diba? NH3. Why ammonia has to be eliminated in the body? Ammonia originates in the catabolism of amino acids. Again, ammonia galing talaga yan sa catabolism or the breaking down of the amino acids that are primarily produced by the degradation of proteins. So parang ang nangyari is naging kanon ka ng protein, na breakdown yan into amino acids. Wala na po itong simplest form pero hihimayin natin siya per functional group. And then dun sa alpha amino group, meron kang na-produce na ammonia. And then mapapansin nyo mamaya sa urea cycle, itong ammonia, meron pa siyang simpler form. Mako-convert siya. Mag-undergo siya ng pathway na medyo mahaba-haba. Okay? But the principle is from the protein to amino acid to ammonia. Because ammonia originates in the catabolism of amino acids that are primarily produced by the degradation of proteins, the dietary as well as existing within the cell. Like what we've mentioned, proteins can be synthesized in our body as well as from our dietary intake. Okay, and then it involves the digestive enzymes, the proteins released by the digestion of cells of the walls of your GIT, the muscle proteins, the hemoglobin, and then the intracellular proteins. Ma'am, bakit po detrimental or masyadong toxic si ammonia sa katawan? Bakit nga ba? Si ammonia po kasi, NH3 po siya, pagka napunta yan sa ating bloodstream, matatandaan, it will gain protons. What will happen to the ammonia if it will gain proton? Nag-gain yan na maraming protons. Magiging NH4 yan. It will become ammonium. Diba? Ammonium na yung tawag sa kanya. Isipin nyo na lang kapag isa kang weightlifter and then madami kang intake ng mga whey protein. Okay, excessive yung protein mo. So it will generate more ammonia. Na-degrade kasi yung protein and then it will generate more ammonia. So imagine this NH3. Excessive siya and then hahalo sa bloodstream kasi nga hindi na kayang i-regulate ng katawan. Sobrang dami na, di ba? So hahalo siya sa bloodstream. So yung bloodstream mo, merong mga protons, nag-gain siya ng proton. What will happen to the ammonium? Okay? The ammonium can pass through and travel papunta sa inyong ating utak. Okay? Pero babanggitin dito 'yon kung ano yung involved na tinatawag natin dito na mga enzymes. But basically, the principle, pag napunta yan sa utak, okay, ammonium siya napunta sa utak, it can cause cerebral edema. Paano nangyari na uminom lang ako ng maraming protein, whey protein, na naging cerebral edema na, ma'am? Yun nga, banggitin natin siya dito. Ammonia is toxic, especially for the CNS, because it reacts with your alpha-ketoglutarate. Okay? Thus, making it limiting for the TCA cycle decrease in ATP level in the cell part na to. Pero pag doon sa ating brain, it can cause cerebral edema pagka nagkaroon ng edema, di ba? Edema, parang mas madami yung water. Magkakaroon ng increase intracranial pressure. Pag mas marami na yung water na nag-attract na siya ng water doon sa brain nyo, mag-i-increase yung intracranial pressure, which is bad, and then it can lead to coma. But hindi po naman directly sinabi ni ma'am na na parami ka lang, napadalawa lang yung bote mo ng whey protein, kasunod nun, cerebral edema, comatose na po. Meron pa rin pong mga other factors to be considered, but basically the principle is ammonia is toxic, ammonia coming from the protein, and the amino acid is toxic, hence it has to be eliminated. Ammonia is toxic especially to the CNS because it reacts with alpha-ketoglutarate, thus making it limiting for the TCA cycle, and then magde-decrease yung ATP level mo. Imagine, pag decrease yung ATP level mo, walang ginagamit yung ating mga cells, okay, as an energy or the metabolic fuel, wala silang fuel to function. So, hindi lang po sa brain may mga effects si ammonia. Liver damage or metabolic disorders associated with elevated ammonia can lead to tremor, slurred speech, blurred vision, coma, and the death. And the normal concentration of your ammonia in the blood, madami na po akong nabasa na references that says 30 to 60 micrometers. Again, normal concentration of ammonia in the blood, it must be maintained at around 30 to 60 micrometers. So, ang dami na nating napuntahan, pero nandun pa rin tayo sa removal of the alpha amino group. So, pagkatapos nun, proceed na tayo with the decarboxylation. It leads to the formation of biogenic amines. These are amines produced by living systems and are important to life. So, ito yung mga example. And then, ito naman po yung mga example natin on how to detoxify your ammonia. We have the reversal of glutamate dehydrogenase reaction, glutamine formation, yung urea formation. Hence, we will be talking about the urea cycle. So, these are some of the ways para ma-eliminate or excrete natin si ammonia from our circulation. And then, ma-maintain natin yung normal blood concentration of the ammonia in our body, which is around 30 to 60 micrometers. And then, the asparagine formation, it is the involvement of aspartic acid plus ammonia, magiging asparagine siya. So again, for the decarboxylation, and then these are the ways of your detoxifying your ammonia. Okay, balikan lang natin, mabilis lang. Again, nandito na po tayo sa part ng ating discussion na kung saan yung mga pathway na diniscuss natin last week, i-discuss na natin yung mga kanilang waste products and how they can be excreted and brought back to the circulation, paano sila mababalik sa circulation, and then regulated on a normal level. So next start tayo sa amino acid. And then sumunod nating discussion yung pinaka chemical structure so that we can know ano yung mga i-breakdown pa sa mga functional groups ni amino acid. So napunta na tayo sa removal of alpha amino group, nabanggit na natin sa oxidative, non-oxidative, and the transamination. So nandun na tayo sa decarboxylation na nabanggit natin. Okay? So nabanggit na po natin, ammonia is toxic to the body, hence it has to be eliminated. And then, these are the ways. We've mentioned the ways on how to detoxify your ammonia. Papasukan na natin si urea formation, urea cycle. So urea, it is also called the Krebs-Henselit cycle. The normal human adult is in nitrogen balance, in a normal adult. that is the amount of nitrogen ingested each day, mainly in the form of dietary protein, is equals to the amount of nitrogen excreted. The major nitrogenous excretory product is what we call your urea. This is very important in the discussion of clinical chemistry, so dapat isapuso nyo na siya. Huwag niyong kalilimutan. Waste product ang urea. The major nitrogenous excretory product is urea, which exits from the body in the form of urine. Again, the major nitrogenous excretory product of your amino acid is your urea, which exits from the body in the form of what? Urine. Urea is synthesized almost exclusively in the liver. It serves as a disposal form of your ammonia, which is toxic particularly to the brain and central nervous system. Normally, the little ammonia present in the blood, the concentration ranges from 30 to 60 micrometers, and then ammonia is rapidly removed from the blood and converted to urea by the liver. Hindi po pwede na magstay si ammonia sa ating bloodstream. Okay, as we all know, yung blood mo, di ba, it can travel to different systems or to different organs in your body. And then it can deliver the ammonia papunta sa CNS. And then it can lead to coma or death. Ito na, pasukan na po natin yung madugong urea cycle. Okay? The urea cycle showing the compartment of its reaction steps in the cytosol and mitochondria. Okay? Yung ammonia, no, si NH4, it will come to the mitochondria. Again, mitochondria. And then it will combine. Okay, magko-combine po siya sa dalawa. Ayan o, dalawang molecule. So, in this process, tawagin na lang natin yung pag-combine niya sa dalawang molecule na dalawang, ayan, HCO3 or your bicarbonate plus ATP. Okay? So, para ma-convert siya into carbamol phosphate. Asan yung carbamol phosphate? Ito siya. Again, balikan natin sa urea cycle. Papasok yung inyong NH4 or your ammonia papunta sa mitochondria. It will fuse with dalawang bicarbonate and then dalawang ATP para maging carbamol phosphate. Pero, hindi po doon natatapos. Para po ma-convert siya into a carbamol phosphate, anong kailangan natin dyan? Siyempre po, kailangan natin ng enzyme. Yun yung tinatawag natin na carbamol phosphate synthetase 1. Again, the ammonia, it will fuse with bicarbonate. Okay? But it needs the enzyme exactly. Pag tinanong sa quiz, ano yung eksaktong enzyme na kailangan para makabuo ng carbamol phosphate, yun yung tinatawag natin na carbamol phosphate synthetase 1. So, pagpasok ni ammonia sa ating mitochondria, ano yung mga kinakailangan niyang molecule para ma-convert siya into carbamol phosphate? So, wag mo nang pansinin yung mga nandyan. Ito po, yung sinasabi ni ma'am. Number 1, kailangan niya po ng ATP. Number 2, kailangan niya po ng HCO3 or your bicarbonate. Next na kailangan niya is si ammonia. So, itong tatlo, makakabuo sila ng carbamol phosphate through the help of your enzyme carbamol phosphate synthetase. Okay? So, this carbamol phosphate, tapos ka na sa number 1, carbamol phosphate, mako-convert naman na siya into a new molecule. Yun yung tinatawag natin na citrulline. Ito siya. Citrulline. But this carbamol phosphate needs an enzyme pa din para ma-convert siya or to catalyze that reaction. And then yung tinatawag natin na ornithine transcarbamolase. So, kailangan po ng ornithine transcarbamolase, okay, para maging citrulline siya. Okay? Nung na-convert na siya into citrulline, kailangan po mag-fuse ni citrulline plus aspartate. Ano yung kanila magiging product? Yun yung tinatawag natin na arginosuccinate. Arginosuccinate. And then, ano ba? Ano ulit ang kailangan para maging end product to para magkaroon tayo ng product na arginosuccinate? Yes po, tama ang nasa isip. Kailangan natin ng enzyme at yun yung tinatawag natin na arginosuccinic acid synthetase. Step number 3. Okay, next step na po tayo. Nung nandito na tayo sa arginosuccinate, kailangan pong ma-convert siya into another molecule. Yun na yung tinatawag natin na fumarate and arginine. And then in order for this arginosuccinate to be converted into a fumarate and arginine, it needs an enzyme that will serve as a catalyst. And that is what we call your arginosuccinase. Arginosuccinase po. Okay, so nung naging fumarate na siya and arginine, okay, naging fumarate and arginine na. Ano na po yung kasunod nito? Medyo malapit na po tayo sa katotohanan. Ang katotohanan na kasunod nun is yung arginine and fumarate nyo, yung arginine natin, it will need an enzyme arginase. This arginase will act upon the arginine and then it will yank or it will produce, yun na yung pinaka-hinahanap natin na urea. Ito na siya. Okay, this urea, ano na mangyari kay urea? It will go to your kidney. How do you shape a kidney? Tama ba yung shape ko? Oh, ano bang meron sa urine? Ay, ano bang meron sa kidney? Diba? It will go to your kidney through your bloodstream and then it will be excreted in the kidney through your urine. Ano po yung sinabi ni ma'am sa description kanina? Urea will be excreted from the body through urine. Pero hindi po sinabi ni ma'am kanina na marami siyang pagdadaanan in the mitochondria bago siya makarating as urine. Ito na yung katotohanan. Again, let us just repeat the urea cycle. Once your ammonia that came from your amino acid enters the mitochondria for the eukaryotic cells, it will combine with two molecules such as your ATP, such as your bicarbonate that needs ATP in order for them to make a carbamol phosphate. And then without the particular enzyme, what we call carbamol phosphate synthetase, hindi siya makakapag-produce ng molecule na tinatawag na carbamol phosphate. So we need this particular enzyme. And then we're done with that step. Step number 1, kasunod nun si carbamol phosphate, it will be converted into citrulline through the help of your enzyme ornithine transcarbamylase. Next, your citrulline will combine or will fuse with your aspartate. Aspartate coming yan dyan sa somewhere. Basta galing yan dyan. Saan po galing si aspartate, ma'am? So, umano lang siya. Parang sumulpot. Hindi. May pinanggalingan po si aspartate dyan. Okay, citrulline plus aspartate. And then, ang kasunod nun si arginosuccinate through the help of the enzyme arginosuccinic acid synthetase. Okay, your arginosuccinate will be converted into fumarate and arginine through the help of your arginosuccinase. And then, pag yung arginine na po, tutulungan siya ni arginase so that it will yank the urea that will go to the bloodstream. For example, bloodstream to. And then, it will go to the kidney. The kidney will be the one who will excrete the urea into the urine. Okay? So, ang sinasabi natin dyan, once na nagkaroon ng mga mutations or nagkaroon ng problem with every enzyme or every molecule, it can be detrimental and it can lead to severe cases or severe diseases. Okay? So, mahalaga na well-functioning ang each and every enzyme inside our body. So imagine kapag hindi mo na-excrete si urea and then it will stay as an ammonia sa inyong katawan. Nagka-problema ka sa mga enzymes mo. Okay, yun po yung sinasabi nating deep importance ni enzymes na dapat hindi siya na-denatured kapag mataas yung temperature sa katawan natin. Kasi itong buong urea cycle na to, it will be parang depleted or parang mako-compromise siya. Okay, so ang haba ng urea cycle. Proceed na po tayo sa amino acids. So amino acids, alam niyo naman niya, ang pinaka-amino acid na alam natin is 20 talaga. But according to some updates, structural formulas and classification of the 21 na daw siya. It includes the selenocysteine. But if you're going to ask, or I will be asking you kung ilan pa ba talaga ang amino acid, 20 pa din. Stick pa rin tayo sa 20. The major function of amino acids as components of the important peptides, amino acid as sources and components, and of important substances. So magkaiba yung importance and then yung uses ni amino acid kay protein. Kasi si amino acid, yun lang yung ginagamit natin para makabuo ng protein. Si protein, sila yung talagang parang basic structure ng ating mga different substances sa katawan. Si amino acid, tumutulong lang siya to synthesize those basic structures. And then the physical and chemical properties of amino acids includes this. Okay? And then there are different methods and principles on how to separate your amino acid mixtures. Principle behind each basis of separation and behavior of amino acids, it can be categorized into paper chromatography, or your ion exchange chromatography. It can be separated through electrophoresis as well. Okay, so ito mga description lang po of the proteins and peptides. Hindi ko na siya i-discuss. And then basically, ito ulit, balikan natin yung chemical structure of the amino acid. We have here our group or the side chain, the alpha carbon, the carboxyl group, COOH. Pag tinanong sa quiz, ano po ang chemical structure of the carboxyl group, COOH? No need to put the sign. Okay, and then the amino group, it can be written as H2N or NH2. Okay, dito talaga nagkakatalo, dito sila nagkakaroon ng main differentiation, yung mga amino acids na pagdating sa R group. Pero pagka lagyan lang natin ng simple hydrogen dyan, okay, meron na tayong nabuo na glycine. That is the simplest amino acid. So these are the non-essential and essential amino acid. Hindi ko naman kayo pagdodrawingin, pero pansinin nyo, same talaga po sila ng structure doon sa mga iba. Nagkatalo-talo lang talaga sa R group. Okay? Meron na po tayong mnemonics on how to memorize the non-essential. Non-essential, ibig sabihin, ito yung mga type of amino acids that can be synthesized naturally by our body. Essential amino acids. This must be incorporated in your dietary intake every day so that our body can use these following essential amino acids and then makagawa ng basic function sa ating katawan. So ito sila. Essential amino acid. Essential po na maisama sila sa ating dietary intake. Yun yung ibig sabihin niya. So these are the classification of amino acids based on the polarity. They are also referred to as neutral amino acids because containing hydrocarbon R groups. The term neutral is used because these R groups do not bear positive or negative charges because they interact poorly with water. Non-polar amino acids play an important role maintaining three-dimensional structure of proteins. So two types of hydrocarbon side chains. We have the aromatic and the aliphatic. So ito yung mga aromatic ring structures. Okay, pakabisado. And then in paper chromatography, the more the branch, the more the non-polar yung amino acid. The more na mas branch yung amino acid, the more na non-polar siya. Example nun, si glycine. It is the simplest and smallest. So it can be accommodated in places inaccessible to other amino acids, particularly where peptides bend sharply. Next, yung proline. Ito yung tinatawag natin na helix breaker, also called in amino acid, and then the methionine, sources of your SAM. Okay, the active metal donor. Ito naman yung mga examples ng ating amino acids with uncharged polar R groups. Bahala na po kayong gumawa ng mnemonics, paano siya kabisaduhin. And they have functional groups capable of hydrogen bonding, so they easily interact with water. Okay? And then the amino acids positively charged R groups, the basic amino acids, LAH. Okay? Positively charged, tandaan, LAH lang po yan. Kung natatandaan si histones, okay, meron siyang lysine and arginine, kaya yung pinaka amino acids involved doon sa histones na laman ng ating mga DNA ay na nagko-compact, nakikipag-compact sa ating DNA sa loob ng chromosome. Yung main amino acids involved in the histones are what we call lysine and arginine, and they are positively charged. Recall lang kasi ang ating mga DNA, they are negatively charged. So positive plus negative, nagkakaroon ng electrostatic reaction. So histidine is the imidazole group responsible for buffering capacity of the hemoglobin. Okay, your hemoglobin serves as a buffer, recall, unique roles in the enzymatic catalysis. So amino acids with negatively charged R groups, dalawa lang naman, madaling tandaan, si aspartic and glutamic acid. So kung nalilito sa positively charged and negatively charged, pwede kayong gumawa ng mnemonics. Basta ako, ang pinakapalatandaan ko dito kapag positively charged, yung involve doon sa DNA, sa histones. Yun, si L, A, tsaka si H. Sama mo na si H. So, yun. Ito, ang mga additional information na lang to. Si glycine, coming from the glycose, meaning sweet, di ba? Greek word po yung glycose, meaning sugar or sweet. And then your peptides with significant biological activity, si glutathione. Ito yung buong pangalan niya, parang Y-glutamyl L-cysteine glycine. It is found in almost all of your organisms. It is involved in many important biological processes like protein and DNA synthesis, drug and environmental toxin metabolism, and amino acid transport. Effective as a reducing agent because the reducing component of your glutathione is the SH group po. Of the cysteine residue, the abbreviation of the reduced glutathione is what we call GCH. GSH. Kasi nga yung SH, yun yung kanyang parang pinaka-reducing agent. So, fun fact lang about the glutathione, it is used mainly as a treatment. Yung mga cancer patients natin, ginagamit po itong glutathione. So, pinaka-side effect lang talaga ni glutathione ang pagputi. Hindi siya talaga yung pinaka-direct agent or substance that causes your skin to be lightened, parang ganon. Side effect lang siya. Mainly, it is a detoxifying agent for your liver. Parang antioxidant siya or parang detoxifying agent nga as what we call. Next, the oxytocin and the vasopressin. Oxytocin, the amino acid sequences of these two peptide hormones differ only by two residues. They both contain 9 amino acids. Again, they both contain 9 amino acids and are produced by the cleavage of polypeptide precursors within different specialized cells in the hypothalamus. Pag tinanong kung saan po nasi-synthesize ang ating oxytocin and vasopressin, they are specialized cells in the and produce precursors in the hypothalamus. Your oxytocin, naririnig na ba? It stimulates the contraction of uterine muscles during childbirth and the ejection of milk by mammary glands during lactation. So in males, oxytocin may have a regulatory role in the synthesis of sex hormone, testosterone. Yung vasopressin naman, it is also known as your antidiuretic hormone. It is secreted in response to low pressure or of your high sodium concentration. ADH acts as a stimulating the kidneys to retain water. So mainly yung vasopressin natin, mainly involved for the control and balance of the osmotic balance natin. Okay, yung blood pressure and regulation ng ating sodium homeostasis. Pagka naman nabanggit si blood pressure, sodium homeostasis, si water retention, asahan mo na involved dyan si kidneys. So ito mga additional information na lang po yan. Ano pa ba yung mga pwede nating i-add na information for the other topics? Nag-iisip si ma'am. Okay. So, we're very lucky na hindi nakasama sa module 13 to 14 yung mga ibang diseases kasama kapag nagkaroon ng mutation dun sa urea cycle, ganyan. Kasi masyado na siyang mahaba and masyado na siyang intensive. So, yung discussion na yun, mas maganda na sa clinical chemistry na siya. And then, same as for your molecular biology. Tama ba? Oo, parang ganun. Meron doon hindi na sinama. Buti na lang. Yung discussion ng talagang mga phenylketonuria, yung mga ganun, pahapyaw lang. Dito pala, sorry, dito yung biochemistry, hindi na siya talagang sinama kasi masyadong madugo. Urea cycle nga lang, baka nababaliw na kayo. Okay? Well, that concludes our discussion for today. It's never too late for anything. Thank you and God bless.